So again, we'll do questions at the end because we'll have the panel come up and you'll have the opportunity to ask questions there. Our next speaker is Lisa Dellafave, who is going to talk about genetics. As one of the things many people in this room share is dystrophin mutations, and she'll take you through that and why it's important to know about that.
Okay, so I'm a genetic counselor. I've been working at the University of Chicago for about 6 years now with MDA for maybe about 10 years on and off, so I've been involved with MDA for a while. So I'm just going to talk about genetics overview. Many of you might already know some of this, for which I apologize. If you don't know it, then hopefully you'll be hearing it you'll get it as we go along. So again, just general principles of genetics, talk about X-linked inheritance, and then how the mutation can affect things. So this is a picture of our cell. I'm going to try and see if the pointer works. It looks like it might. This is a picture of a cell. This is the nucleus inside. This is where all of our genetic materials contained. You can see it looks kind of like little squiggly spaghetti, and that's kind of what it looks like if you look at it under a microscope. And then on the outside, this is important where all of our structural components of the cell are. And so when we get to dystrophin, that's what I'll be talking about too. So this is a picture of if we split open a cell, what our DNA actually looks like. It does kind of look a little bit like spaghetti. Each of these individual pieces is a chromosome. And if you actually go ahead and line them up, which as a genetic counseling student, we had to cut them out piece by piece and try and figure out where the 2 number 1s were and 2 number 2s were, and it was not a fun process, and then you sneeze and you just get really angry at yourself. So what we are mostly concerned with is this X and this Y down here. So males and females carry the same first 22 chromosomes, and then this would be a male with an X and a Y. Females have 2 X chromosomes, so that's how we can tell if it's a male or female. So what does a chromosome do? Basically, our chromosomes hold our genes. So there's thousands of genes on every single chromosome. And then our genes are what make proteins. And proteins are actually what function in our body. And proteins are the things that when a muscle's not working right, it's going to be a protein that's the cause. But if you actually reverse backwards, it's really the DNA that's the underlying cause there. So in Becker muscular dystrophy, this protein here, is that working? Okay. This protein right here is the one that's causing the problem, the dystrophin. And you can see it's a huge protein. Where'd it go? And it actually links our outer cell membrane with our inner cell, like the muscle cells that are on the inside. And so if dystrophin's not working right, you can kind of picture how the cell just might not be as strong as it should be, because that's actually the linking thing that links that whole cell together. So this is kind of what I was talking about. What does it do? Well, it's in the cytoplasm. It provides some reinforcement for the cell, and it links that extracellular matrix, the outer part of the cell, to the whole, or the extracellular matrix on the inside to the whole cellular process on the outside. And what's the cause of Becker muscular dystrophy? Well, essentially that dystrophin protein's not working the way that it should. It's shortened when it should be a little bit longer, or it's not, or it's structured a little bit differently than it should. So this is actually if somebody's had a muscle biopsy. We're just going to kind of quickly look at on the right-hand side. For a muscle that has a normal amount of dystrophin, you can see how it's really the outlining of the cell is really bright. But for somebody who has Becker muscular dystrophy, it's not outlined as brightly. So that would be a reduced amount of dystrophin. And then for somebody who has Duchenne, they actually have no dystrophin. So for Becker, we're seeing some dystrophin, which is why it's less severe than something like Duchenne, because there's still some dystrophin there to link some of those cells together. It's just not quite enough that we need for that muscle to be working normally. So getting back to genes, and then we'll come back to dystrophin. If we look at our actual genetic code, it's made up of A's, C's, T's, and G's. And I'm going to talk a little bit more about what these letters do. But there's 4 letters that make up our genetic code, and they link together. And if you probably have all seen this deoxyribonucleic acid, the DNA shape. Well, this is actually how you get the shape. The C's and the G's link together, whoops. And then in the end, it actually looks like the little spiral of DNA that we all know and love. So basically, our DNA contains these things called exons and introns. So one gene has a whole bunch of exons. The exons are the ones that actually code for the protein. And then the introns are kind of in the middle of the exons. And for a long time, we didn't think introns were important, but now we know that they actually might be a lot more important than we thought. But we're still learning more about exactly what they do and how important they are. So when we make our kind of thing that goes in between the protein and the DNA, which is called mRNA, the introns get spliced out, and the exons just go one right after another, so they line up. And then the protein gets made off of this thing called mRNA. And then that would be the full-length protein, or dystrophin protein, basically. So if we have a change in our DNA, then that can actually change the protein, because now you've seen how the DNA goes to the protein. So if there's actually a change in the DNA, you're going to get a change in the protein, because the protein just copies whatever the DNA says. So here we're back to our A, C's, T's, and G's. And within each gene, they're basically lined up in a very specific order. Now, because we're all different, some of us have very little changes within our DNA, and that's what makes us all different. So sometimes in the genetic testing world, it can be really frustrating if we see a change and we're like, is it because we're different, or is it actually because it's meaningful, and that's what's causing the disease? For the most part in Duchenne and Becker, we can kind of tell that what happens is actually a disease-causing change. There's not as much variability that makes it a little bit more troublesome for us. The vast majority of people with Becker are going to have a deletion of exons. Is this sounding familiar to some of you who have had genetic testing? You know what exons are deleted. You can rattle off, 46 and 47 or whatever it is. So basically what happens is, if these are our exons lined up, and again, the Duchenne gene's one of the biggest ones out there, so there are a lot of exons in the gene. So I just picked a couple here. But basically, if somebody has a deletion, say that you have 44, 45, 46, 47, 48, well, maybe their 45 and 46 aren't here anymore. So they're just going to have 44, 47, and 48. We can actually tell that on genetic testing. So this is what the most common person with Becker is going to have. But other things that we can find is, for instance, what's called a well, let me go back to the normal here. You see our A, C's, T's, and G's all lined up. Underneath these letters are the proteins. That's what kind of lines up to make our protein. So one of the important things is 3 letters make one protein. So when we're trying to reveal the whole genetic code, we always go by 3 letters to figure out what protein it's going to be. So say, for instance, this C here, which is just a C, doesn't look that important, right? But if it actually gets changed to, sorry, I'm trying to keep the pointer going here. If it gets changed to an A, well, that could actually be a pretty big problem. It doesn't seem like it's a big deal, but this TGA could actually cause that, what we call a stop codon, or basically our genetic material at the very end, there's this very specific codon that says, stop, I'm done. We don't need to do anything else with this gene. Totally done. It should be at the end. Ideally, that's where you want it. But if a mutation or a change happens right in the middle of a gene, it could actually produce that stop codon way before it should end. And so if you've heard about people with stop codons, that's actually how one forms. So basically, once the whole translation happens, and if it hits one of these, it's going to think, I'm done. But it doesn't realize, wait a minute, you've got all this other gene left, and it doesn't know the difference. And so these people are going to truncate their protein right here. A missense mutation is basically a change from one letter, a C to a G, which actually then changes a protein. So it should be this protein, and instead it's this one. That can actually alter the shape of the protein and how it functions. Those are a little bit less common in Becker. So this is kind of the breakdown. Dystrophin, like I said, is a humongous protein, 79 exons, which is one of the biggest genes out there. The vast majority of people, about 85% of people with Becker, are going to have deletions and duplications of exons. Some people can have small insertions, those point mutations that I was just talking about, changing a C to a G. And then about 15% of people with dystrophin mutations are going to have stop codons, premature stop codons. So some people think, well, I have a humongous deletion, and so that's why it might be more severe. Well, a lot of times it's actually the size of the deletion that doesn't matter as much, but what it's actually doing, again, to the reading frame of the DNA. So it all comes back to the DNA. So here we have our 3 letters grouped together again. If this is a deletion here in the middle, and if the deletion ends right at the point where the 3 letters picks back up where it should be picking back up, it's called in frame. And that means that everything, it gets deleted, but it actually might be able to pick back up and keep reading this protein. And since it's going to make normal protein, because it's still, those letters are still placed exactly where they should be for somebody with a normal dystrophin gene. And so that's considered in frame. Those people are most likely going to be Becker patients because they're going to have the vast majority of their protein that's still considered normal. The difference is, if there's a deletion here, a big chunk missing, and this, say, A goes along with that deletion and it picks up at the T, you're always going to come back to, you need 3 letters, and so it says, okay, well, I'm just going to take this TCG. Well, that's going to make a totally different protein. So everything from this point on is going to be out of frame. It's just going to make what is going to be considered gobbledygook. It's not going to be able to see it. And then that protein actually is just not going to be functioning well at all, or it'll wind up being degraded. And the cell just doesn't know what to do with it, and it says, I have no clue, get it out of here, which means that you're not going to have any dystrophin left. So these people are going to be the ones with Duchenne, that protein's not present. So I just wanted to really quickly run through, you guys have probably heard of some of the new clinical trials going on. Many of these are more so for Duchenne people because they're trying to make people with Duchenne more like Becker's. So these are like some of the exon skipping and some of the stop codon read-through trials. So just the quick basis of these are, if these are our exons again, right here in the blue, then typically if you have a stop codon again, it's just going to stop reading early. With stop codon read-through, the premise is that there's a chemical that can actually induce a change where that mutation, or where that stop codon was inserted, so that the protein can just keep on going instead of stopping. So that's kind of the premise for that one, to hopefully get more functional protein into these people. Exon skipping, the premise is that if people don't have, if they've got, say, exons 49 and 50 deleted, then it's to induce this 48 to meet up with this 51 to get more protein made. And in frame is the important thing. So remember, most of these people have out-of-frame mutations. So this slide will actually show it a little bit better, which is actually an interesting way to think about it. So these are all the different exons in Duchenne. Like I said, there's 79 of them. It's huge. say that somebody has a deletion in exons like, I have to make sure I'm doing this right, say 45 and 46. So these two are deleted. Exon 44, you can see at the end here, it's a blunt end. Well, that means that this one has to link up next time with a blunt end. If it links up at like 51, it's not going to be able to, the gene's not going to be able to still be created because it has this arrow here. So in exon skipping, it's kind of targeting that medication to make sure that two ends that meet up can really meet up together and trying to keep that protein going on. So just to give you guys a little bit of an idea, because it's all over the, Duchenne and Becker websites, and so to let you know what it means. So now we're going to talk a little bit about inheritance, in case you haven't heard of that. So when we pass our chromosomes on, if you think back to our genes, you remember the 2 number 1s, 2 number 2s, 2 number 3s all together. We pass our chromosomes on so that we only pass on one of each, so that half comes from mom and half comes from dad, and when they come back together to make an embryo, you've got the full 46 again. So basically, half go in one egg or sperm, the other half go in the other egg or sperm, and come back together. So in X-linked inheritance, we're again looking at these two little chromosomes down here, our X and our Y. Again, females have 2 X's, males have 1 X and 1 Y. So males are affected because they only have 1 X. The dystrophin gene is on the X chromosome. So if the dystrophin gene is affected, they only have that 1 X. They don't have another one to compensate, whereas females do because they have 2 X's. So one of their X's might not be working right, but they have their other dystrophin protein that's working just perfectly, which is for the most part why the vast majority of females are unaffected, because they've got that other X to kind of keep that protein at a good level that doesn't get them into trouble. So for those females who are carrying mutations, again, the vast majority of us don't know that we're actually carrying mutations. If the green X is the one with the female, and that's the X with the mutation, she can either pass on that X, and then her spouse passes on his X, because he only has 1 X to pass on. So this child, for instance, is going to be a carrier, because she has 1 green X and 1 normal X. But that child would be unaffected. Or mom can pass on the black X that doesn't have the dystrophin mutation. That would be another girl, because the man's going to pass on his X, so that's going to be unaffected, not a carrier either. Or she can pass on her X with the mutation, and then dad determines the sex, right? It's all up to dad what we get. So this time dad's making a boy, but she passed on her X with the mutation. So that person, actually I have them backwards. Sorry. That person's going to be affected. And then mom could also pass on her X without the mutation. Dad'll make another boy. Sorry, that line should be over a little bit. And he's going to be unaffected. So basically it's 25% chance whether the male's, 25% chance with each pregnancy that it will be a male and will be affected. Now for the males who actually have Becker, if they want to have children, basically they have their 1X. So essentially they determine essentially all of their daughters are going to be carriers because they can only pass on the X. And their X, if they're going to make a girl, is going to be the one with Duchenne. So 100% of the females will be carriers, but they will not have any males who are affected because they pass on the Y. In order to make a male, they got to pass their Y on. Does that make sense? All right. So this is a typical pedigree of X-linked inheritance. You can see, oh, rats. How did I do that? Hold on. Okay. Oh, perfect. That was easy. So if we have a carrier female here, This is basically the 25%. You know, we've got 4 kids here, so she can either have a son who's affected, a son who's unaffected, a daughter who's unaffected, or a daughter who's a carrier. And then if we've got her brother, and this is the same thing, the son is going to pass on, all of his daughters are going to be carriers. Okay, give me a second. Let me, I froze.
Oh my goodness,
I know.
that's weird.
Hold on.
Do you have a lot more? I'm gonna have to restart.
Oh,
Sorry.
it's okay. Let me think what I had next.
Sorry.
That's okay. Actually, you know what? Let me just, let me quick go grab my slides, because then I can just talk through the rest. Those were the important ones with the pictures, so.
Hold on. Sorry about that. It'll just take a minute.
Well, I guess it died at a good time, because I didn't really have any more pictures. So, yeah, I'll do it. So basically about 1/3 of boys with Duchenne and Becker actually have a mom who's a carrier, and then 2/3 of moms are, I'm sorry, 1/3 are not carriers, 2/3 are carriers. And so basically it's still a pretty high, like, what we consider a new, what we consider a new mutation rate, because it's about 30%. And then moms, even if they don't have a mutation within their blood, there's still what we consider germline mosaicism. Essentially, it means that within their eggs or within their ovaries, there actually might be a little population of cells that also still have the mutation in there. It's what we call germline mosaicism. It happens about 14% of the time. And so when women still, if they were to do the genetic testing in their blood and they're negative, we still say there is still some residual risk of about 14% that it could just be a small population in the eggs that might carry that same mutation. So as far as, I just want to briefly touch on, if you haven't had genetic testing, how you can go about it. Basically, talk to your neurologist, or if there's a genetic counselor in your clinic, that's the other easy way to go, and you can talk to them about it. It's a blood test, so that's the easy part. It gets sent off to a lab. About 4 to 6 weeks later, it comes back, and we wind up seeing whether they could identify the actual deletion that's present. We need to start by testing the person who actually has Becker muscular dystrophy instead of testing a family member. That's because whenever we do genetic testing in any disease, you always have to start with the person who's considered affected to make sure that you can detect the mutation. Because remember, genetic testing still is not 100%. We wish it would be, but we're not quite there yet with technology. So you always want to start with the person who has it. If on the off chance that the person who has it is unable to be tested, we could maybe start by testing other female relatives, but it's really not an ideal situation. We really, really, really want to start with the person who actually has Becker. But then that information can then be passed along to other relatives. So why is it beneficial? Well, for instance, if there's other women in the family, sisters, aunts, cousins, whoever, who might be thinking of having children, they might want that information. So things that would be important are we might want to monitor their heart a little bit later on in life because, again, although the vast majority of females are not affected, there are actually quite a few carriers out there who can have some heart palpitations, heart arrhythmias, and even late onset cardiomyopathy. And so we really do want to make sure that they're being followed appropriately and are recognized as being carriers so that they get that medical management. That's really important. The other thing is that people who are of childbearing age might consider prenatal diagnosis. That would be like an amniocentesis, or we can now do pre-implantation genetic diagnosis, and that's actually before the woman gets pregnant, you can fertilize an egg and a sperm in a petri dish and pull off just one little cell at the 8-cell stage and test that for the deletion or whatever mutation it is that we're looking for. And then we only implant those embryos that are unaffected. For some people, they look at that as an option that they want to explore too, but we need the genetic testing result in order to be able to do that. And then obviously there's lots of new clinical trials coming down the line, and so many of those trials are requiring mutation, whether we know the mutation or not. So you can get geared up for that and be ready by doing genetic testing now. And then for those of you who don't know what genetic counseling is, it's basically just we explain to patients more about what the diagnosis is, what the genetics is, kind of what I did today. Take a family history, talk to them about different options for testing or other things in the family, and also provide resources and act as an advocate. So, and that's my contact info. That's about all.